webinar of our Translacore network for the early career investigators. And today we are having Kelly BRUNEL from University of Montpellier. She's a second year PhD student in Alexandre David team. Yeah, she's going to present some results of her work. Kelly, the floor is yours. Oh, and a presentation is going to be recorded. That will be for submission to the web. You know, you will have a chance to kind of like edit some minutes.
Okay.
Presentation. Please mute your microphones so that we're not disturbing the presentation. Okay, Kelly.
So thank you, thank you for the introduction, and to give me this opportunity to present my work today, which is about the use of epitranscriptomics for the detection and management of pancreatic cancer, that I'm conducting in Alexandre David team at the IRCM and its Cyberna company, and under the supervision of Stanislas Quesada and Amandine Bastide. So the pancreatic cancer represents a major public health concern. The incidence is still increasing, and it's expected to become the second leading cause of cancer death by 2030. It's a very aggressive cancer, and still we have a very poor prognosis with a 5-year overall rate under 10%. The causes are multifactorial. First, this cancer is often diagnosed very late because there is a lack of robust diagnostic biomarkers. It also relies a lot on its microenvironment that helps the tumor to progress. So it's very important to better characterize the interaction between those different cells of this microenvironment to better understand this pathophysiology of cancer. And also it's a very heterogeneous tumor with a few therapeutic targets and a lot of resistance to the conventional therapies. So for all these different challenges, we believe in the team that the epitranscriptomics could bring new solutions. And I'm going to focus on PDAC, which are the pancreatic ductal adenocarcinoma, because they represent the vast majority of pancreatic cancers. So the epitranscriptomic is all the chemical modification of RNA. We can find them in all subtype of RNA, and for the moment, there is already 170 RNA modification that have been identified. They are regulated by a different type of enzyme, the writers that had the modification to the RNA, the eraser that removed it, and the reader which will interpret it. So it has been shown that those modifications could impact some key post-transcriptional process, such as the RNA stability, the splicing, but also the translation efficiency that at the end could influence the cancer evolution and adaptation. This is something that is well documented and well known. For my project, I will focus on a more innovative aspect of those RNA modification at the nucleoside level. So the nucleoside is the building blocks of those RNA. And for a long time, they were considered as biomarkers of byproducts of RNA degradation with no function. Until recently, an article that came out showing they have biological activity. They could bind to some receptor and influence human and inflammation response, but still this hasn't been studied in the context of cancer. And those modifications, those nucleosides, the unmodified one, they could be recycled by the cells to form other building blocks for transcription or into end products. But it's not the case for modified nucleosides, which are not recognized by the enzyme of the cell-wash pathway. So they are systematically excreted out of the cells, which makes them really easy to measure in the bloodstream, for instance. So knowing that, my working hypothesis is that the circulating modified nucleoside could represent new non-invasive biomarkers for the diagnosis, for the prognosis, and therapeutic purposes of PDAC, but also that they can maybe act as cellular mediators and this way influence the tumor microenvironment remodeling. So first, I'm going to introduce you how this epitranscriptomics could be used to identify new biomarkers. So in the team, we have established a pipeline to study the epitranscriptomics. It's from patient samples. It could be cells, tissue, or plasma. We extract the nucleoside and we analyze them in liquid chromatography coupled with mass spectrometry. And then we use some multivariate analysis and machine learning algorithm to identify robust epitranscriptomic signature that responds to a clinical need. So I'm gonna present you some results I got from the BCB cohorts that were composed of plasma sample from 32 pancreatic cancer patients. And I also included some sample from healthy donors to identify some diagnostic and prognostic biomarkers. So the second step is to extract the nucleoside from the sample. So it's basically a metabolite extraction based on methanol. Then we deplete the samples of lipids to avoid its dirty, let's say, the liquid chromatography system. I didn't have the word, sorry. And we dry them and resuspend them in the mobile phase of the liquid chromatography. So then we can inject the samples. They will be separate based on the chemical properties, such as the polarity. Then we ionize the molecules so that they can be detectable by the mass spectrometer. These ions are filtered based on the mass to charge ratio. So in the team, we have established a method that enables the detection of more than 60 nucleosides for the moment. And then we fragment these ions and analyze them again to another mass spectrometer to increase the sensitivity and specificity of the method. So at the end, we got a chromatogram, and for each peak, each compound is represented by a peak, and 8 represent their relative abundance in the samples. So then we can do some analysis. So what I've done first is to compare the epitranscriptomic profile I obtained from these two populations to, at the end, identify some diagnostic biomarker. So first I use PCA to get an overview of the distribution of this patient according to the whole epitranscriptomic profile. So as you can see, we have a clear spatial distinction between those two populations, meaning there is some clear difference of epitranscriptomic profile based on the type of patient. So then I generate a heat map to easily visualize the biggest difference in nucleoside level between the population. So basically in the row, it's all the nucleosides that were detected. They are anonymous because at the end, Siberna company wants to develop an in vitro diagnostic test. And you have all the patients in column, and color ranging gives information about the fold change compare the two populations. So when it's red, it's that the nucleoside level is increasing in the pancreatic cancer population. When it's blue, it's decreasing. So from this heat map, I choose 6 nucleosides, which are the most significantly expressed between those patients, the ones with the red arrow, and I combine them to create this diagnostic signature. And then I wanted to test the classification performance of this signature. So I use some work curve. And as you can see, I obtained AUC score of 0.988, which is very close to 1. So it shows that this signature has a really good sensitivity and specificity. So I identify a diagnostic signature, but then the objective would be to validate this signature in another cohort, an independent one and bigger one with more samples from early stage patients. But also I would like to use to identify early detections by biomarkers signature. So I would use the PACTO cohort, which is based on some benign lesion of the pancreas, such as the acute pancreatitis, but also some lesions which are precancerous lesions. There are a risk of degenerescence into PDAC, such as the mucinous cystadenoma. The objective is to have a biomarker that would indicate if we're in the benign lesion or if it's already a risk of degenerescence or there is already a cancer. And in this way, we could also see if the previous signature that I identified is really specific of the carcinogenesis process, or if it's more broadly associated with pancreatic pathologies. I also wanted to see if this signature is specific of pancreatic cancer. So I compared the level of the nucleoside from my signature with other cancer types, such as the glioma or the colorectal cancer. And as you can see, we see that there is an overall similar trends for the cancer, except for the nucleus 12. So we are developing calibration curve to have an absolute quantification of the level of the nucleoside, because for the moment we only see relative quantification, so we can only see trends if it's increased compared to the healthy donors or it decreased. And this will allow us to set up threshold, clinical threshold for the detection of this cancer. At the prognosis level, I wanted to see whether we could use the nucleoside as an indicator of the survival of the patients. So from the early stages pancreatic cancer patients, from their profile, for each nucleoside, I separate the population into 2 groups, the low median group or the high median group. And I compare their survival probabilities using Kaplan-Meier curves. So from all the nucleosides that I detected before, there were 20, only 2 stand out. And they are nucleoside number 4 and nucleoside number 12. Those results show that the patients which have a higher level of this nucleoside basically have a poor survival. So this indicates that probably those nucleosides could be involved in the evolution, the tumor progression, or even confer some aggressiveness of the cancer. So they represent very good prognostic biomarker, but still this has been obtained on a very few amounts of patients. So this result needs to be confirmed. Okay, so I don't know if I go directly to the second part, or if you want to stop. Okay, get question at the end. Okay, so now I will introduce you about how the epitranscriptomics could influence the tumor microenvironment, and we can see if those nucleosides could have a signaling action. So first, it's important to define the PDAC microenvironment. So is composed of a lot of cancer-associated fibroblast CAF cells. It represents 80% of the tumor mass. And so this the lot of CAF produce large amount of extracellular matrix. This induce a desmoplasia reaction, and it really form a barrier to drugs. So it contributes to resistance to treatments and to make this microenvironment very hypoxic and poorly vascularized. It's also a microenvironment which is rich in immunosuppressive cells, and it participates to the human inflammation and limits the anti-tumor immunity. So my goal is to understand, better characterize the crosstalk between those different actors of the tumor microenvironment with the focus on the nucleoside, modified nucleoside. So we need to know which type of modified nucleoside are secreted by which type of cells, and if those secreted nucleoside will affect the nearby cells and mediate perhaps intercellular communication that will at the end reshape the tumor microenvironment. So to do that, at first I wanted to study the nucleoside secretome of the cells. Remember, I told you that the modified nucleoside are systematically excreted out of the cells. So in vitro models, such as cancer cell line, it's really easy to detect those modifications in the culture media. So that's what I've done, following the same pipeline as I described before. And so I quantified the relative abundance of nucleoside present in the culture media of pancreatic cancer cell line. And so in the cells, there is an abundance of unmodified nucleoside. In the extracellular compartment, we have much modification because there's more modified nucleoside which are detected. And what's interesting is that if we cross those results with the ones I obtained from the patient cohort, so from the plasma samples, it's like the majority of the modification we found are in common. And what's interesting is that the most abundant one, the ones that it seems to be really in high amounts, they're also the ones which are the most abundant in the plasma sample. So it's most likely like this modification comes from the tumor cells. But I also checked on human cells and stromal cells the same way, the nucleoside level in culture media. So M0, it's undifferentiated macrophage. It's for human cells. You can see it seems like they secrete some few modifications preferable in preference. There's more of this modification, but for CAF, which are stromal cells, there is no preference in the secretion of some nucleoside. It really seems that the cancer cells are the ones that secreting in high levels certain nucleoside that could affect all the cells, but this will we will also use absolute quantification to really can compare different cell lines. So it really seems like the nucleoside secretome is cell line dependent. Okay, so now I wanted to see whether the secreted nucleoside from the pancreatic cells could influence the signalization in other cell line. So I told you in the introduction that it has been shown that some nucleoside could bind to receptor. It has been shown they could bind to some adenosine receptor. Those receptors are expressed in a lot of human cells. They are GPCR. There is 4 subtypes. So there is A1 and A3, which is GI coupled. So basically, they will inhibit the adenylyl cyclase, which is responsible for the conversion of AMP into CAMP. And there's also A2A and A2B, which are GS couples, so they will activate it. Basically, the A1 and A3 promote more a pro-inflammatory phenotype, whether the A2A and A2B promote an anti-inflammatory response. And in the context of cancer, it's well documented that adenosine activate those kind of receptor and participate to the tumor immunosuppression. So I wanted to see whether those modified nucleoside could also activate those receptor and lead to a tumor immunosuppression phenotype. So initially focused on the modification derived from adenosine, so on M6A and AAM, which are single methylation modification, and M6AM, which is a double methylation of adenosine. So I work with Le Bon Goûter team, IDIGF, to set up a system to measure the activation of this receptor. The idea is to transfect some THO cells with the receptor of interest and to measure the CMP, which is produced in response to stimulation with some adenosine or nucleoside. And so we add CMP coupled with a fluorophore. It will compete with the CMP, which is naturally produced by the cells, to bind to some antibody, which is also coupled with a fluorophore. So when these two actors are close, it's produce a signal because there is a transfer of energy, and this signal is easy to measure. So we can convert it at the end to a CMP production level. So I use some controls, the adenosine itself, which is the curve in blue, and the NECA, which is another agonist of those receptors. And on the right, you have the curve, which corresponds to the different modified nucleosides. You can see that they are able to reach the same maximal response as adenosine. So they act as full agonists of those this receptor, but still the EC50, it's shift to the right. So it means that we need more nucleoside amount to reach the same half-life, half activation. But what stands out from those results is M6A, which is the brown curve here, which is very close to what we got with adenosine. So from this result, I've been able to show that, yes, some modified nucleoside could bind and activate those receptors. So we know that when those receptors are activated, this will lead to anti-inflammatory response, and it could participate to this immunosuppression. One mechanism is to increase the polarization of macrophage into an M2 subtype, which is an immunosuppressive one, in comparison to the M1 subtype, which are more pro-inflammatory macrophage. So because those macrophages are also the dominant human population in the PDAC, I first wanted to see the impact of nucleoside activation of adenosine receptor on the regulation of those macrophages. So I set up a model of macrophages, and I quantified the level of those receptors on the different cells. So it seems like there is a lot of A2A receptor, which is expressed on M1 cells, and a lot of A2B, which is expressed on undifferentiated cells. Knowing all of this, my hypothesis is that some modified nucleoside, by binding to this A2A receptor on M1 cells, could favor a switch into M1 to M2 phenotypes. So to check this hypothesis, first I wanted to see which signaling pathway could be activated in response to CMP increase. Because it's a second messenger, we know that it could activate PKA, which is also in response triggers some signaling pathway activation, such as AKT and CREB, which are key players of cell proliferation, survival, and also human cells recruitment. So I treated M1 macrophages with different amount of adenosine or M6A, which is a nucleoside, for different times. So here, basically, it's M1 cells. Here you have adenosine for 10, 30, or 60 stimulation. M6A with 10 micromolar or M6A 1000 micromolar. So we can see that after adenosine treatment, we have an increase in the level of CREB phosphorylation, which is peaking after 10 minutes. For M6A, it seems like the activation is more late. We have the peak at 30 minutes, which is also dose dependent, as when we increase the dose, we have more activation, more phosphorylation of CREB. Regarding AKT, it seems like adenosine does not affect the phosphorylation process, but M6A seems to activate it really rapidly. So this increase of phosphorylation protein seems like there is an activation of this signaling pathway. But to be sure of that, we need to look at the gene expression at the end. So I evaluate the expression of some cytokine, pro-inflammatory cytokine, such as TNF-alpha. And I've shown that with adenosine or NICA, we have a clear than expression of this cytokine. So it seems like these CREB activations lead to a decrease of this level. But we know that TNF-alpha is its target of the NF-κB pathway. So my hypothesis is that maybe the increase of phosphorylation of CREB compete with the activation of NF-κB because the co-activator of this pathway are already taken. So next, the goal would be to also expose stromal cells with modified nucleoside, and in the same way evaluate the impact on the phenotype and the function, and also use a more physiological model, let's say, which is a multicellular model comprising tumor cells, calf, and human cells, to also evaluate the impact of those nucleosides on human infiltration. So thank you for your attention, and thanks for all my collaborator and the team.
Thank you so much for your presentation, Kelly. Really clear and really great. I don't know if Alexander, I think he was able to join us, so I don't know if he would like to add something.
I can first apologize because I thought I had a meeting at the hospital before and I thought I could connect from there, but hospital network is is not really good for Teams or Zoom meeting. So the connection was very bad, so I had to take the car and come back as soon as I can. So thanks, Kelly, anyway, for this great presentation. It's a very complicated, ambitious project, I would say, because one part is very highly clinical, with like very defined objectives, and we have very good, interesting preliminary data. But the molecular part is requires a lot of setup, and I think we are almost there. I think Kelly almost optimized, fine-tune everything. So now, I mean, soon enough, we'll be able to dig into the molecular mechanism and see whether immune cells are affected, or even like other type of cell, tumor cells, fibroblasts are affected by surrounding modified nucleosides, because we do see significant changes in terms of concentration. So they must do something. It cannot be just a waste.
Exactly. This is really interesting, actually, if I may take an advantage of asking a first question before. So if anybody wants to ask a question, please either raise your hand or post a question in the chat. I can just begin, whilst others are thinking. I was thinking about the transporters, because if you in terms of release of nucleosides, it's the changing level of SLC, for instance, would be important in that aspect. And another thing that might be change the composition of the extracellular nucleosides would be also the aspect of hypoxia. So I wanted to ask whether you looked hypoxia itself could be, for instance, causing bigger overlap with what you see maybe in the plasma of, in between in vitro cell cultures and the plasma from patients, maybe, it would be more corresponding. Do you have any experience with that? And then the transporters level might be different, yeah.
So I checked in the literature. There are some papers showing that in PDAC, there is some of these transporter which are overexpressed. So it could also explain why we have more modified nucleoside, but it's not the only causes. There is also the enzyme responsible for this modification, which are often overexpressed or down-expressed. So it could also explain why in the extracellular compartment of these pancreatic cells, we have more modified nucleoside. So yeah, it would be very interesting to also look at that. And for the hypoxia, yes, also it's well documented that for adenosine itself, there is a lot of adenosine because of hypoxia. But for the moment, I didn't look at the direct effect of hypoxia on the release of those modified nucleoside in the media. But at the end, I want also to look at the co-culture and this spheroid model, so which will contribute to hypoxia environments. But yeah, that would be clearly on the list of things to explore.
Yes. Thank you. That's super cool. Are there any questions? Yeah, hypoxia might also be impacting the whole immune signaling, what we observed in some other studies. It has an impact on the presentation of TGF-beta receptor, also on different cells and signaling through that, and that might also impact the whole immune environment there. So that would be really interesting to look at that also under hypoxic conditions. So yeah, and I was wondering, because the dynamics of the response that you're following, you've seen changes after 10 minutes, half an hour. So from your perspective, if it's something happening over a longer period of time, days, we're talking here, yeah? And would you expect that, constantly you have a higher level of the extracellular nucleosides that are already present there? So do you think that it would be something that would be interfering with whatever kind of signaling would be triggered by something else? Or do you have any experience with longer time follow-up of exposure to adenosine?
Okay, so I didn't look at days. I didn't treat for days for more than some hours because I add in the media the nucleoside itself. So it's for the Western blot signaling pathway. I want to see something which is very quick. But I forgot what I wanted to add. You were saying about the
longer time follow-up, because I've been just wondering how to do that, because it came up to my mind that there might be extracellular enzymes, because I think it could be then modified to I don't remember, hypoxanthine, no, something other types of nucleosides, and then so the matter of stability, yeah, how stable is that? And then can you observe longer time effects of like, you know, what that would be?
Yeah, maybe the nucleoside they could have an impact by themselves, but also they could have an impact by disrupting this adenosine signaling, which is already present. There will be very interesting to look at that, but for the moment, I didn't have an idea of the relative amount of these nucleosides, so I didn't try to put them all together. When we will have absolute quantification that would be able to do, we will really make a mixture of all those nucleosides and see really their impact on this signaling. But I know that for adenosine, it's very quick, like it's a signaling which is really tightly controlled, and so there is adenosine produced, but it's really rapidly disappearing.
Okay, great.
Maybe another aspect she didn't talk about, but obviously because of the GPCRs are highly expressed in neurons too. We'll also, of course, do some screening with an institute nearby to see whether they could be involved in that, in neuron signaling. And then, you know, there is now this rising field where neurobiology and immunology are, you know, like are connected, talking to each other. So, I mean, this would make sense, like in this regard. So that's also something we are trying to explore. But the thing is, it's just the beginning. So, I mean, we don't know what to try. We try a little bit everything, but yeah, I think like Kelly suggested, the best would be to try to fit to the maximum to what you can see in vivo, because otherwise, then I mean, at some point, if you use a lot of any kind of molecule, you will get something, but is it really relevant? It's really hard to tell. There is a question for you, Kelly.
Yeah.
Hello, Kelly.
Hello.
Thank you for this nice presentation. So, a naive question maybe, I was wondering if polymorphism in adenocyte receptors have been correlated with the prognosis in these cancers.
I don't have an idea. Yeah, I don't know. That would be interesting to look at.
Yeah, thank you.
Are there any other questions? Well, if not, then I guess we will be finishing. Thank you so much.
Thank
Thank
you.
you for the presentation. I'm really happy you could give a talk today, and it was really interesting, fantastic. Good luck with your project, and
Thank you.
thank you all for joining.
Thank you. Have a good day.
Good day.
Thanks a lot.
Thank you very